we can start. Good evening, everyone, and welcome to our monthly webinar, which has been organized by the Institute of Public Health here at College of Medicine and Health Sciences at UAE University. I'm Dr. Mariam Bashir, an adjunct assistant professor at IPH, and today we are going to talk about Biobank and Genome Projects, international benchmarking, analysis, and lessons learned, which is going to be presented by Dr. Jacqueline Hall. So this webinar is actually one of the many webinar series that is being organized by the IPH in order to explore emerging public health issues or challenges, and also to invite different renowned experts from across the world to come and share with us their cutting-edge projects and research. It is actually a part of the global campaign, This is Public Health, which is affiliated with ASFA, that is the Association of Schools of Public Health in the European region. So before we proceed, I would like to just highlight a few housekeeping notes. Please post your questions and comments in the Q&A chat. You can see the option at the middle bottom of your screen. And you can do so while the presentation is going on, and at the end we'll try and cover as many questions as possible. Just to remind everyone that this webinar is being recorded, and the recording will be posted at our YouTube channel, that is the IPH YouTube channel. This webinar is being accredited by the UAE University as 1CME, and at the end of the webinar, we will share the link to the evaluation forms with you. Kindly fill out those forms in order to obtain your CME certificates. Okay, so it is my absolute pleasure to introduce to you Dr. Jacqueline Hall, who is a pioneer in genomics, healthcare, and technology with more than 2 decades of experience. And she is currently a consultant and has founded her own genomic platforms company, that is the Vivactive in the United Kingdom. She is also an honorary senior lecturer at the Imperial College London. She holds a PhD and postdoc in bioinformatics and machine learning, and has an extensive background in genomics and clinical research, having played a vital role in several large-scale genome sequencing initiatives and genomic platforms across the world. So Dr. Jacqueline Hall actually has a vast experience, including being the head of translation research at the European Organization for Research and Treatment of Cancer. And she worked with genomic platform companies across the world and collaborated with Genomics England on the 100,000 Genomes project. Her work has significantly contributed to the integration of biosamples and biomarkers into precision medicine clinical studies across Europe and beyond. As a visionary leader, Dr. Jacqueline Hall continues to inspire and drive positive change, translating novel technologies to clinical applications, thereby revolutionizing healthcare. I hereby welcome Dr. Jacqueline Hall to present to us today on Biobank and Genome Project, international benchmarking analysis and lessons learned. You're very welcome, Dr. Jacqueline, and the floor is yours.
Hey, thank you. I need to press the share button. Here we go. Hopefully this is showing.
Yes, it is showing.
Good?
Yeah, that's great.
Great. So thank you very much for the kind introduction. It's a pleasure to be here today. I'm very happy to be invited to speak to you all and to talk about some of the experiences that I've had working in this field. And I know you did a very thorough introduction just now on career, but I did want to take a slightly different angle on this, just on one slide just before I begin, because I find it very important to explain also to those people who are deciding, what studies they want to do or what jobs they may want to consider in the future, that there's a lot of different opportunities out there. And I took a rather unusual career path for someone who trained in bioinformatics. So I just wanted to take a moment to talk about a couple of points in my career, I think, which have really opened up new doors for me. And these are points that I would thoroughly recommend others to consider if they're thinking about a career in this space. And so originally I trained in biochemistry, but early on I realized that I liked all the biology and the science, but working in the lab was not really something that suited me. It wasn't my passion. My passion was the data and the information coming from the biology. So I had an opportunity to do a PhD in cancer bioinformatics, and I didn't really know much about what cancer bioinformatics was, but I had taken the opportunity in my studies as an undergraduate to do an industrial placement year with GlaxoSmithKline. So in this year, I worked for the company full-time in between my second and third year at university. So I was there for 1 year, and I had a great mentor and boss, and he introduced me to in silico programming. So this was an internship where we were programming to predict whether or not a drug would be toxic in, excuse me, preclinical animal models. So an early screening to understand whether drugs that they were developing would be safe for human use before even taking it into lab tests. And I didn't even know this world existed, but I spent a year working on building in silico models, and I realized that actually there's a whole area of computational biology, computational chemistry that opened my eyes. So I went back, finished my last year at university, and the opportunity of the cancer bioinformatics PhD was there. And I said, great, I get to do the science. I get to work on a topic which I'm passionate about because people in my family have also been subject to cancer, unfortunately. And so I wanted to do something, in healthcare that had an impact. And I thought, okay, this is my chance. I can learn how to do science with the computer. And that really led me on a very interesting path. I did do a postdoc in Canada for a couple of years, and that was working on breast cancer, again, using the bioinformatics skills. But at that time, I realized it was very difficult to get hold of the data that we needed in order to build the models that we wanted to do. So we were trying to predict what is the right kind of treatment to give an individual based off the expression profile of the breast cancer. And we had some data sets that we were using, and we were using public data sets as well. But we just didn't have the right data, the right clinical follow-up information, and all the pieces that we needed to really build good models for this kind of predictive work. So I actually went to work in a clinical trials organization in Europe. It was a group that I'd been following for some time. They have a big name in doing cancer clinical trials. They're a pan-European organization running clinical trials in Europe. And they do a lot of standards and setting of standards in this area. And I went to see them, and I said, look, I'm actually a bioinformatician, and I know that you guys do clinical trials, but I think that it would be really great if we could work together somehow, because you sit on a lot of data. You have all these different clinical trials with cancer patients being enrolled, where the biosamples are being collected, and in some cases, they are generating molecular data on the biosamples, biomarkers, even they were starting to do sequencing at that time. And they had the clinical data from the clinical trial. So they were following the patients to see what treatment they had, what outcome they had, did they respond, was there a good quality of life experienced by these patients. So this was a fantastic opportunity in my mind that we could actually start to build some of these data sets where it would lend itself to do predictive modeling in the future. And they said, great, we don't have any bioinformaticians that work here. We have plenty of statisticians who run the clinical trial design and all of this, but bioinformatics, we don't have. And so I ended up working there for 6 years. And this was really a fantastic opportunity to get into the whole world of translational research and what is the infrastructure and quality standards required to put all of these operations in place. And we were collecting biosamples, so we had standards for the biosample collection, and marrying that with the information from the clinical trial. And importantly, we set up processes for secondary research use. So the clinical trials would have an objective, looking at a particular drug or a particular procedure. But of course, once you collect the data, other questions could be asked of that data, also going across multiple trials, like meta-analysis and this kind of information. So we set up processes that other researchers in the field could apply to use either the samples to generate more data or the data that was already existing for further research. And so this is what led me more into these large-scale programs and the coordination and management required and the governance which is required in order to manage these programs. So at this point, I wasn't really doing much bioinformatic programming anymore, as you might imagine. And after that, because I had been working extensively with different European organizations, and I was living in Brussels at the time, I had good contact with the European Society of Pathology, and I helped them out on a couple of projects. But I saw that they were doing this great work around proficiency testing, where samples are sent to a laboratory for testing, where the third party knows what the right answer is, but the lab doesn't know. So they receive the sample, they return the results, and the third party tells them if they got it right or not. So this is what proficiency testing is, and it's used to ensure that labs' clinical practices are maintained and quality is maintained over a period of time. I had never heard of proficiency testing before I started working in clinical trials. It's a very specific area, but again, it opened a whole nother world where I understood actually there's a lot more you can do with a bioinformatics or biochemistry degree than I had first imagined. And at that time, we sought funding from the pharmaceutical and diagnostic industry to help support this initiative. And so again, this was another eye-opening experience where we were working alongside the companies and alongside oncologists and pathologists from around the world who were really expert in this field on how we could help put these processes in place to support labs. And after that, I got very much into the space of genomic sequencing, because a lot of the labs were starting to adopt this in clinical testing, and it was a novel technology coming forward, and I'm lending my bioinformatics skills on that, or interest, I should say. I thought this was an up-and-coming area that I would like to get interested in, and I worked for a number of different platform companies, so Seven Bridges Genomics, also an interpretation company, so GenX do whole genome sequencing interpretation. And then I ended up consulting for, and then working with IQVIA, who are a large data and clinical trials company. And they were working together with Genomics England to understand how the 100,000 genome data could be used to support drug discovery and drug development with the pharma and diagnostic industry to really accelerate how we can get better treatments to patients faster. And that was a really wonderful experience. I learned a lot from that on the interface between research and application in drug discovery. And since then, I've been consulting under Vivactive Limited, which is my UK entity, and actually had a wonderful opportunity to consult on other national genome programs as well, including some in the Middle East. So that is my quick story about how I ended up where I am. But for anyone who, also younger people who want to talk about careers or whatever, I'm always very passionate about sharing my expertise. So feel free to reach out to me. I'm happy to talk about that with you in person. So to the topic, Biobank and Genome Programs. So today I wanted to cover a quick introduction to genomics, and then to position a little bit an introduction on large-scale genome programs and why these have sort of popped up around the world in many places, because they have a number of benefits associated with them. To give a couple of case studies, and from the learnings that we get either working on the program or studying other case studies of how different countries have approached this or different initiatives have approached this, I sort of accumulated a kind of model of best practice on how to manage large-scale genome sequencing programs, which is sort of a personal viewpoint from my experience, but also bringing in experience from the European frameworks where information is shared about these national genome programs. And then come to lessons learned from some of these large-scale programs, what went well and what has not gone quite so well in some cases, and how we can apply that in the future to be successful with these programs. So as you probably well know, the cost of human genome sequencing has been and is declining quickly. So even in 2022, one company announced the ability to do the $100 genome. So the falling costs have made this technology so much more accessible for clinical practice and also for research. So more data is arriving on the scene using genome sequencing. And there's a couple of different types of sequencing. So just to quickly run through this, the different options are you could sequence an entire genome of someone, so their inherited DNA, and this would be covering all of the genes on the non-coding regions of the DNA. Obviously, this takes a longer time to do than some of the other methods and is generally the most expensive one. But even the costs for whole genome sequencing are starting to come down now. And especially if you do large-scale sequencing, you can get, in the past, this has definitely been a major driver in the field, is having economies of scale. So having very large sequencing units where you fill it up with many, many samples, and that way you can bring the cost down. This is slightly changing, and I will come on to that a little in a further slide. Another option is, instead of looking at the whole genome, we could just look at the coding genes only. So this would be the exome, which is about 1 to 2% of the total DNA. This is cheaper than doing the whole genome. And the third option is to just look at selected genes. So for example, maybe you have known clinically validated risk genes for breast cancer. You could just do a panel on breast cancer genes and look at someone's genome and report these. So this is obviously a much smaller proportion of the genome, depending on how many genes you're looking at, but could be like between 10 and 500. And this can be very rapid and also much cheaper. So this could either be done using next generation sequencing technology to make a gene panel and report that, or there's other methods like microarrays and SNP arrays that can be used to generate this kind of data as well. So different options, obviously they give you different amounts of information. And another thing I wanted to cover briefly is a new thing that we're seeing now in the genomic space. So when we do genome sequencing with next generation sequencing, the model is that we break the DNA up into small pieces to then do the chemistry and understand the base calling within those different pieces, but we then align those. It's almost like putting it together in a book, putting the pages back together to make a book. We align them to a reference genome, so like a human reference genome if it's a human sample, for example. And then we do the variant calling to understand which ones of those are disease causing or natural variation, or ask whatever question it is that we're asking about the data. So normally, typically for most of the older next generation sequencing technologies, these were always making short reads. So they were only able to cover like short fragments of the DNA when they were reading the fragments. So we have new technologies coming through now. So for example, Pacific Biosciences or Oxford Nanopore, where actually they're able to read much longer pieces of the DNA. So this actually makes it easier to put all the information back together to understand the core genome, and has certain advantages in terms of the types of variation or the type of base calling that is being generated from the data. So in Oxford Nanopore sequencing in particular, one of the benefits for this is it's actually using a very, very different way to sequence the DNA. It actually has a pore, and the DNA flows through the pore and it looks at the electric current. And then from this, it's in finding out what the bases are for the DNA, which is very different from some of the other technologies where it's using chemistry to identify those. But one of the advantages of the nanopore sequencing is this technology is very portable. So they even have handheld devices. And you can put the samples in and get the data coming real time. So you can start the sequencing, look at the data as it comes from the machine, and then stop it when you have enough data, or start it again, stop, start, as you like. And so there's also this possibility to do adaptive sampling, where you tell the machine that you want a certain coverage or really take a lot of depth and information on this part of the genome, but on this part of the genome, just do a light coverage with, light information. So you can adjust the machine to get the depth of information that you want as you go along the genome. The other methods don't have this kind of flexibility. So this is very new and exciting technology. And the reason that I'm mentioning this is it's changing a little bit how the field is working in terms of organizing large-scale programs. So in the past, you need to have the big machine, fill it with a lot of samples for economy of scale. But now with some of these new technologies where you get very rapid data, samples can come in and be sequenced straight away, you get the data straight away, and you can do a light data, keep it if there's something interesting, go back and then look in depth and run some more on the same sample in a very flexible way. So this has led to the ability to have more decentralized sequencing. So there's lower capital costs to get started. And in fact, it lends itself to sequencing anytime, any place in a very portable and scalable way. So I wanted to then, with that backdrop, talk a little bit more about the national genome programs that we've been seeing around the world, what genomic initiatives there are. There's different types, and I'll give some case studies and examples on those. And in the past, a lot of these national genome programs or these large-scale databases would either be driven by the either by SNP array, so that's the very small panel type of sequencing or array. Because of costs, that was the cheapest way to do a large volume of samples. So a lot of these programs took that approach in the beginning. As the costs came down or some specific applications became of interest, more and more programs started moving to either exome sequencing to get more of the DNA or even full whole genome sequencing. But obviously that's a very expensive option when you're doing a large-scale program with a lot of people. And so most of these different programs around the world in the past have been focusing on using the short read technology. But some of the newer programs, as this new technology has come along, they've been adopting some of the new sequencing as well, or even doing a combination of long and short read and putting that together because they tell you slightly different information. And so, for example, Genomics England, initially the 100,000 genome study, this was done with Illumina sequencing. And the purpose of Genomics England was very much focused on delivering whole genome sequencing. And I'll give the use case in a minute. And so their economies of scale was very important to centralize everything. Other programs, for example, like in UAE, there's been an adoption of using both long and short read sequencing as these new technologies became available. And I think in the UAE, they're one of the programs with the largest volume of nanopore sequencing data in the world currently in the database. But as you can see from this graph, a lot of the sequencing efforts and a lot of the activities has been happening in America, not so much in some other parts of the world, but also in Europe. Some of this activity has been publicly focused and some is more private from companies. But there's a lot of reasons why governments and agencies have become very interested in these large-scale genome programs, because it can bring a lot of benefits, not only to the individuals for improving precision medicine, for example, so getting accelerated medical research and improving personalized treatment. So for cancer patients, what's the right treatment for them? Or for rare disease, getting better diagnosis using the genetic data, but also potentially for prevention of disease, so looking at risk factors in the DNA. But it can also lead to a lot of cost savings for healthcare systems because you get the right drugs to the right people and with less side effects or problems, and also can have the opportunity for early intervention if there are people with high risk to save them from later problems. So overall, it can lead to a healthier population and more productive workforce, but economically, it has benefits as well for creation of skilled jobs. So, and it avoids people going to another country potentially to study something if you have an initiative at home where you're able to stay and study what you want to do. And obviously, by having such high-tech research, it can stimulate novel innovations and foster collaboration with companies, industry, and has the potential to create interesting intellectual property and knowledge. And in fact, there's been a couple of reports out there to say that precision medicine has the potential to create hundreds of billions in savings. And that was a report from McKinsey in the US, but also that genomic research itself can add a lot to the economy and all of the jobs and everything that's associated with that. So just to give a couple of examples of how sequencing can support clinical applications to help support disease diagnosis and prevention. So we could be looking at someone's hereditary DNA, and this can help us to diagnose rare diseases, which sometimes can take a very long time to diagnose without this data. So that speeds that up a lot, and then they can get the appropriate treatments that they need. It can be used for risk. So for example, looking at people's risk for cardiovascular disease, and then follow those individuals with a specific program to help prevent any heart attacks or cholesterol problems. And that's a preventative type of approach. Also, another application is pharmacogenetics. So this is looking at an individual's DNA and seeing what are the best drugs to give them that will not give them any side effects, or adjusting the dose of the drug depending on the genetics of the person to make sure that they're getting the right dose, not too much, not too little. There's also lots of applications for sequencing cancer tissue for understanding what is the right drug to give the individual based off the tissue sample. And for this, you need quite a rapid turnaround time to be able to know what is the right treatment to give to the cancer patient. But there's lots of other applications in the areas of infectious disease. We saw a lot of sequencing in COVID, for example, but also transplant, where maybe you need to match an organ from a donor with an individual. And so sequencing or genotyping can help make the best match possible for that person. There's a lot of examples of how genetic data and genomic data can be used for drug discovery as well. And I don't want to go through all of these examples, but it can range from understanding areas of need, disease burden, the mechanisms driving the disease. So if we can understand these properly, including genetic data, then we know how to create the right drugs. It can also be used for efficiency in clinical trials and clinical trial design, and identifying the right patients to go in the clinical trial as well. And it can be used for things like drug repurposing. So if a drug works well in one indication where there's a specific genetic profile, and then you find that genetic similar profile in another indication, then it could suggest that that drug could actually be useful in more than one circumstance. So I just wanted to say a quick word. So this is quite a famous example. So some individuals, they lack a functional copy of both genes. So everyone has two genes, one from your other, one from your father. And some individuals lack the functional copy of both of these. So this is called a natural human knockout, but they may not show any particular physical ailment or noticeable phenotype. So this was seen, for example, with healthy people, where they had a knockout of this ApoC gene, or APO3, where the family members, they had to drink a fat-laden milkshake, and it was shown that actually, when they didn't have this gene, their fat levels stayed the same, where normally you would expect it to spike. So this was a very interesting finding that demonstrated that if we could get drugs to target this particular pathway in this gene, that it would be very effective for controlling fat and cholesterol, but without any side effects. So that's an example of how genetics can be used for driving drug discovery. So there's a lot of different types of national genome programs and biobanks out there. As I've alluded to, some have been more focused on disease. So rare disease and cancer has been a big topic, and looking at genetic data on how to tackle those diseases. Also population-based programs. So this is more all comers. So sequencing an entire group of people, whether they have disease or not, and then understanding what risk factors might be important for that population. There are other initiatives which have been very biobank and sample focused, where they have strong clinical data. So for an example of that would be UK Biobank. So although we call it a biobank, it also has a lot of genetic data. So my definition of biobank and genomic program is not very distinct from each other because they overlap very closely. But the UK Biobank, their focus was to collect samples and collect very deep clinical data for people over a long period of time, 30 years plus. Since then, they've done a lot of sequencing to annotate that data as well. So now it's a genetic database as well as having the sample and the clinical data, but it was primarily driven by biobanking and clinical information. Whereas some other initiatives have been very much genomic data driven. So starting with generating a lot of genomic data and then annotating or focusing on bringing in the clinical data. Some programs have been more research focused, and some programs have been very much more about clinical implementation. So how do we actually get clinical genetic testing into the healthcare system? And in fact, some of the programs have also been a mix of those. So Genomics England, for example, has very much been a mix of both a research project and clinical implementation of services. And as mentioned, some of these programs have been government-led and some are more from companies. And so this can vary. But one trend that we are seeing generally in this field is federation of data. So now we have these different databases coming. The question is, can we put them together? And how do we make sense of the larger data set? And this is something that's been very active in Europe, for example, with the Beyond 1 Million Genome Program, where the different national genome programs across Europe, and I have a little diagram here, have come together to actually share data and also generate new data. So this is really about networking of data, and this raises a lot more questions around, do we have the right quality? What are the standards for sharing data so that we're comparing apples with apples and not trying to put apples and oranges together? So this is a definite trend, whereas in the past, people have just been generating a database themselves and it sits there. But now there is very much a trend towards networking. So a couple of examples. I've talked a little bit about Genomics England already. So they started with the research program of 100,000 individuals. And the topic here was really looking at how to implement whole genome sequencing. And it was one of the few programs that really went out there to do whole genome sequencing in the beginning. Many of the other programs, because they wanted like larger volumes of participants, they went for more cheaper options like panels or SNP profiling or exome. So what Genomics England is set up is owned by the Department of Health and Social Care, so a government initiative, and it's adjacent to and linked with the NHS. And the idea was, let's take the difficult cases from the NHS, either rare disease cases like where we don't have a proper diagnosis, or cancer patients. That those people are not having genetic testing or don't have adequate genetic testing within the NHS already, because we have some clinical genetic testing, a lot of clinical genetic testing already happening in the NHS, which is quite straightforward and simple and everyone knows how it works. Let's take the hard cases and do whole genome sequencing and see if we can work out what's going on. So it's about 85% of these cases were rare disease and the rest were cancer. So this allowed them to build a research database adjacent to the clinical setting, but also have the opportunity to return results to patients where something was found. So for example, a rare disease patient without a proper diagnosis was coming from the NHS, get a whole genome sequence. If they could identify like a proper diagnosis and gene, this information would then be reported back to the clinical service for that individual person. But at the same time, they had this database of 100,000 genomes with associated clinical information, which is coming from the medical records of the participants. And that is there also for research purposes. So they have collaborations with companies to access this data and understand how it can be used for drug discovery purposes. And they have collected a lot of data. It's now much more than 100,000. I think it's more like 150,000 that they have. A different example in Finland, so this has been very much a focus of a research program, but it has a strong public-private partnership aspect. So they created a consortium working with a number of different companies, but also the Finnish government and universities. And they focus very much on a biobank, so collecting the sample and the health data. And then they added the small amount of SNP data from genomic sequencing. So as in they didn't do full genome sequencing on this, they just did the SNP array. But this allowed them to 500,000 individuals. They're almost at their quota now. And they're working with 13 different companies. So this is a very good example of how working together with industry has been successful in bringing together this program. And they had a lot of new findings. It's a very specific genetic makeup in the Finnish population. So they've done a great job at looking at that. So a third example I wanted to mention is Precise Singapore. So this is a national program in Singapore, and they've taken an approach of 3 phases. So the first one was more like looking at a healthy population. Let's see what is normal for an Asian population in terms of genomic variants, and let's establish capability. Let's look at how the economic modeling is going to look, and work with some industry partners in the first phase. And they do 10,000 in the first phase. Then they scaled up to the 100,000 part of the program, where they really wanted to include more genetic diversity and both healthy and disease individuals. So they expanded that out. And then this is looking more at how can we use it in clinical implementation, training the workforce, linking in more clinical data. And then the third phase is the going big phase. So that's going up to 1 million genomes. And here they even want to link social and environmental data and look at cost-effective analysis on a much larger scale. So they're kind of covering both healthy and disease, but in different phases of the program. I mentioned this initiative. So what's also really important when we're looking at different populations and we're trying to interpret what does the genomic data mean, is that obviously we're not all the same, and we have different genomes, and in different parts of the world, normal looks different, right? So a lot of these programs have integrated parts of the work around what is a reference genome for our population, because they may have higher rates of some variants and lower rates of others. And this has been a fundamental part of how we can improve our understanding of the genome going forward. And it means that we need to take care of things like diversity and equity. So lack of diversity in genomic research can limit our understanding of the human genome, and it's essential for us to understand more for us to get the biology right. And also from an equity and ethical responsibility point of view, it's important to have different people's genomes represented in the database. And as you can see from this graph, it's certainly not equally distributed around the world. So there's some new programs which are also coming out. So for example, the Caribbean Genome Program, which is being run by Carry Genetics. And here we're using nanopore sequencing. So I should also say I work with this company. I forgot to mention that earlier. So here we're using nanopore sequencing. So it's very scalable, but we're able to do capacity locally. So there's a lab in Bermuda, but we're going to train other labs across the Caribbean on how to do the sequencing so that the samples don't have to be sent away. And this can also increase not only the diversity of the data, but increase local capacity and jobs for the Caribbean. So the goal there is to implement genetic testing locally for clinical purposes. There's also consumer offerings, so health and wellness type tests, but then also to support clinical research and the large-scale genome sequencing program and reduce sending the samples away. So key elements of a large-scale genome sequencing program. I wanted to just briefly touch on this and come to some successes and learnings about how we implement these programs. 5 key pillars include having the right ethical and legal governance framework, how we engage the populations and the clinicians and the citizens that we're working with in these programs. Obviously, we need to have technical infrastructure and standards in place in order to execute on these programs, but we need to build expertise, workforce, and local capacity to be able to deliver the results back and have all the necessary follow-on and clinical interventions where needed. And all of this needs to be done in a way which we can scale, because we don't want to start a program and then have a problem when you scale it up to, let's say, 100,000, the turnaround time or the database isn't large enough. So everything has to be planned to be able to scale and for it to be sustainable. So some of these programs, even if organized as a research project, you don't just want the funding to run out at year 3 and stop. We want these databases to live on, to have extra data to be used to, also impact clinical care where possible. So, as we talk about legal frameworks as the first pillar, over the years, I sort of put together a little maturity framework on some of the elements that we look for in an environment to know. It shows how mature the environment is for doing a genomic program, for example. So some of these indicators are things like, is there a well-developed governance framework for data? So data privacy legislation, or, in Europe, we have GDPR, for example. Some countries even brought out specific genomic legislation. Other frameworks around biobanking and sample collection, even if they're not legislation, but guidelines, this also indicates sort of maturity towards being able to deliver these programs. If there's any policy or governance around reusing samples and how ethics approvals take place, this is a strong indicator of a mature framework. Looking at things like electronic medical records is a strong indicator of a mature area, but also things like quality policies, health economics policy, and whether or not there's any genetic discrimination laws. So these are some indicators that can tell us whether the environment that we're working in is ready for a big genome program, or whether there's some elements that we still need to work on, or when we deliver the program that we need to be very clear on what these items are going to be in terms of local policy, even if there isn't a legislative framework in place yet. In terms of the technical infrastructure, we have to think about sample collection, so biobanking, the sequencing itself, the laboratories doing that work, how we analyze and interpret the data, and that has been a bottleneck in several programs because it's been underestimated the work required to actually interpret this information. We need a place to store the data. We need to have good security around the data to make sure it stays safe. But genomic data on its own is not that useful. We need to have it linked with the medical information and to be able to go back to the samples to generate more data in the future. And this also, as part of the technical infrastructure, we need to have clinical organization and standards around this, so appropriate data models, harmonization, and guidelines to support how we do all of these things and what the quality metrics are. So that's a key point. Of course, we need to engage people. We need public awareness. We need if there's patients involved, we need their acceptance. The clinicians need to be motivated and engaged, and so we have to work with them and train them. They might need extra training on how to deliver the genomics or what to do with the results. International harmonization has become a big thing now, as I mentioned, with networking, but also how do we engage with industry around the data? Training capacity and workforce is a critical point, and it can be slow to train and have enough people ready for the program. So this is definitely something to think about early. And that can mean even getting people interested in doing this as a career. So career awareness, degrees, education for younger people, but then also for qualified medical professionals, getting them up to speed on the latest genomics, or for doctors, for example, how they should treat the results, and then monitoring this capacity over time to make sure that we still have enough people to deliver. In terms of scalability and sustainability, we have to think about how is this being funded? Is this public funding? What happens at the end of the public funding? Is there going to be test reimbursement so that the clinical testing becomes part of the healthcare framework? How would the database be supported 5 years from now? Looking at some of the cost effectiveness analysis, Australia has done a really good job on that, as have some other places as well, but looking at the benefits from sequencing people early that allow us to justify spending the money on the data creation for later benefits, and then benefit sharing as well. So some common challenges, I kind of alluded to a lot of these earlier in the framework, but bringing together this aspect of research and clinical care, it's kind of the key, but it's also quite difficult to do because research works in quite a different way than clinical when you have a patient sitting in front of you who needs a result. So putting those two things side by side can be a challenge, and they're working on different time frames and to different standards sometimes. So that has been a common challenge for many of these programs, especially the programs who focus more on research. Then they have the problem, how do we actually get this in the clinic? So I think one of the lessons learned there is think about both the clinical application and the research at the same time from the start. Having the right maturity framework, as I mentioned earlier, thinking about these different policies or governance models or legislation that needs to be in place so that people feel safe when they join the program, that we know what we're working to for the data standards, who can have access to the data, how and when. Having this clear from the beginning, even if there isn't a specific legislative framework to have a policy for the program, and an implementation plan for the program is absolutely key. But having political support and engagement from governments or ministries is also really important. But raising awareness with the public and communication with the public about what this program is, how they can get involved, and what the output of that program is really, really important for maintaining trust with the public as well, and for getting the program started with having people enroll. Some programs have seen technical issues regarding infrastructure. So for example, not starting with cloud and then realizing the data is very big and that now we need to work with cloud, or having long turnaround times for reporting results because there were just not enough clinicians who were trained to be able to properly look at the data and sign off the reports, or a lack of use of tools to facilitate that process of variant interpretation. So that's something that's been encountered with some programs as well. But not only educating the clinicians, but educating policymakers, those who are making decisions on training about what is genomics and the benefits of genomics. And a key point, I think, if you want to make a sort of long-term sustainable program, is working together with industry as well. And that needs to be thought about carefully from the beginning. And I think I've covered actually all of those examples because I know that we want to save some time for questions. So I kind of put together my 5 key ingredients of what I would think is a framework for a successful national genome program or large-scale program. So financing is obviously key, and sustainability around that financing, having the right infrastructure, having political will and support in the environment for that, the right regulatory data governance framework, and also having a dedicated coordinating entity who can actually execute on this, who has experience and knowledge of some of these successes and failures and how to manage these programs. Because we could have the best idea in the world, but we need to actually put it into practice for it to take shape. So that is also a key part of execution. I just wanted to say a last word here around what we're seeing now is a trend also because of the way that genomics is changing. And I mentioned that it's becoming more and more decentralized. We have this technology which can be adopted relatively quickly in a scalable fashion, that addressing this data gap around diversity and having benefit sharing and equity-based principles around genomic data is becoming more and more topical. This is an interesting article that came out quite recently, and they just also were reiterating that having representation of different groups, even within the same population, different ethnic groups or social demographic groups, this is important. We need to think about how we can include these people in science and reduce any marginalized populations and mitigate bias. So who has benefits, how people are engaged, and promoting equitable science is definitely a key theme going forward. And so I think we're going to see more policy work on this in the future. So watch that space. Great. Well, I think we probably want to save some time for questions. So I will stop the presentation there.
Yes, thank you very much, Dr. Jacqueline, for this very interesting and very informative presentation. Thank you for sharing your experiences and also the lessons learned from different genomic programs across the globe. Thank you for that. So we have a few minutes for questions. Before the audience is ready, I think I would like to ask two questions, and of course, it's kind of public health related. I just want to know, like, what are the collaboration opportunities between public health and this genomic programs, and how do you think these programs have impacted advances in public health?
Yeah, no, that's really interesting. So I think there's lots to do. So are there potentials for collaboration? I think the short answer is definitely yes. So I kind of alluded to this a little bit, I guess, in the presentation, but we can talk more about it, is this aspect of there's kind of a research part of genomics, because we're still understanding what the human genome is, what's normal, what does it mean if there's different variants. So that's definitely part of the sort of science realm. And then there's the clinical realm of how do we actually apply this for, you know, disease or for prevention. So at a sort of population or public health level, there's that application. And I think that crossover of going from, let's say, a scientific finding to something that's clinically relevant, it takes too long currently, right? We need better ways to speed up our learning and demonstrate that we have robust information which is ready for clinical practice from our understanding of the human genome. And international collaboration, including diversity in the data, is very, very important for us to be able to do that and to do that better. Because if the databases and what we're learning about genetics and genomics isn't representative of everyone, when we go to a public health level, we're not going to do a good job for everybody. So there's definitely that sort of scientific translation into clinical application. And then on the other side is around the economics of that, right? So how do we make that sustainable? Obviously, I mentioned that some of these sequencing technologies are now changing, the costs are coming down, the way that we do sequencing is changing. So I think it's really important to look at the cost effectiveness and have mechanisms like things like health technology assessment, where we can bring these new technologies and evaluate them and their cost benefit information to be able to apply them in a public health setting. So because if we don't capture that data or we don't have a good mechanism to evaluate that, we're going to get stuck again, because we'll be like, okay, we've got this great new technology, but it's too expensive, or we don't know how to apply it, or we don't know what the benefit of using it is in the public health setting. So it would probably get stopped. So I think those are two on the science side, but on the economic side, two key ways that I think there's a lot to do in terms of how we can facilitate those processes and speed that up.
Okay, thank you very much. We have someone raising her hand, Nahla Kazim. If you can unmute. Yes, please.
Okay, yeah. Thank you, Dr. Jacqueline, for covering such a fantastic topic in a simple manner. And from my side, as a clinician, we are trying to integrate the genomic testings for screening the population, while we as healthcare providers and clinicians, we are gearing up for implementing this, you know, and putting it into our practice. My concern is that we are not simultaneously putting the public awareness at the forefront of what we are doing. So my also frustration is that we don't have any universal pattern of counseling patients to do the genomic testing.
Right.
Or yeah, for the patient's education, or counseling the patient, or even consenting, taking the consent from the patients that what we are doing and how your tests are going to be used and for how long. So there is lack of the way we do our practice. There is lack of approaching the patients themselves. I need your input into this thing.
Yeah, so I'm trying to think of some concrete examples, and I might actually have a little bit on one of these slides that I skipped over. So it's a long process to do this kind of public education. So there's education at the level of citizens and public, that they kind of get an idea of what is genomics, and they're not like completely afraid of it. Because you know, if they go to the clinic for something and someone says, "Oh, I'm going to test your genome," they're like, "What?" You know, so there's even that level. Then there's of the patients when they come in, to explain to them, okay, well, this is a test that can help us do X, Y, and Z, either find you the right treatment, or can be used to prevent something, or so that explanation and that piece of education. So one method that has been used is around working with patient advocacy groups. So let's say it's about rare disease, working with a rare disease advocacy group, and then have them really help, like campaigns and so on, about how genomics can help in that disease. That's something that we've seen quite a bit of, and can be quite powerful. Also, I wanted to mention this. So in Estonia, the Estonian Biobank and Genome Program there, they had a really big focus on public engagement. So it might be interesting to dive into more detail on some of the things that they did. But they did a really wide-ranging communication plan over years. They had things on TV, they had newspapers, social media, they even had a story in the soap opera, which included something about genetic testing, just to familiarize people with what it's about. And for them, they felt that having that emphasis from the beginning was super important, because it gets transparency, it builds trust and engagement. Other countries, so Qatar also did quite a bit of work on identifying sort of literacy of genomics in the population. So did people kind of understand what it was? And also some campaigns around, you know, how it can be used. But a lack of genetic counselors, on people who can understand, like explain to people what the data means, is a worldwide problem. There are some tech solutions which are coming now, which might help. So some of these AI tools that can help answer the basic questions on genetic counseling, and then someone talk to a proper counselor, rather than the counselor spending like half an hour explaining what is a gene, what is, you know. So there is some ways to sort of streamline that, but we do need more people to do training. So building this workforce is super important as well, and getting people excited about going into this field, which is why I wanted to spend a little bit of time in the beginning to sort of talk about career paths and so on, because it's a really exciting area for people to work, and we need to find ways to engage people to be willing to come on board and do the training.
Thank you very much, Doctor. I think we have a few more questions in the Q&A and the chats, but due to limited time, we'll just take one or two. So this is saying from Hiba Al-Balushi. Can you elaborate on the governance of European Biobank integration? Maybe just briefly elaborate.
So I guess the question is about integrating the genomics and medical records.
Yes, there is that also. There is that also. Maybe you should take that one because will be more concise.
Yes, so having the genetic data on its own, there's only limited questions and understanding we can get from that. So it's so important to have additional data sources attached to that information, so that we can make correlations between health, disease, and different genetic variants. And I guess one important thing for UK Biobank, they initially started with collecting the samples, and it was healthy volunteers, but over time, obviously, some people got sick or, you know, had problems, but they kept in touch with those individuals, and, you know, they also were collecting additional data, either additional samples or medical record information, and following up on their health status. So one of the key things I would say about a successful program is it's not just about building the database, it's about building engagement with the people who sit behind the database, because not only you want to keep them engaged in your program, but it's important to have access to them for going back to ask more information, or collecting another sample and finding out what happened. And that enriches the data so that the data stays living, because if you put the data in a drawer and leave it, it will go off, right? So it's very important to have additional annotation being added to the data all the time. And in many ways, actually generating the genomic data, if we do a whole genome, you only do it once. but the clinical data is the hard part because it changes over time and you have to keep updating it.
Thank you. I think there are two very important questions. Maybe I just say one or two things about them. One is the role of AI in genomic programs and healthcare. And then the other is on if you've had any ethical issues from your experience or in your company.
Yeah. So role of AI, there's a number of different applications where AI can help. So AI can help from an operational standpoint. I kind of gave an example already for the genetic counseling. So having, you know, conversational agents or chatbots which can educate patients or help patients understand their data before using the time of a clinician or a genetic counselor, or who can, the chatbot can maybe help answer questions about the study, or, you know, from an operational perspective, there are ways that those types of technology can be useful. But also from understanding the data, obviously there's a lot of applications for the use of machine learning and AI in understanding the genomic data and the linkage with health information. So a general trend that we've seen in the past, we had to have a massive database to build a big model to understand the relationships in the data and make predictions. This is kind of going on its head now. So with some of these new AI tools, the focus is actually, let's just collect 25 people, but we'll get really deep data on those 25 people. And this is sufficient for some of these models to find the right associations and so on, to then have predictive value. So we're seeing quite a lot now in that space, because it's much more efficient to collect 25 people, even if it's deep data, than to collect hundreds and hundreds of thousands of data. So this is a big change actually that AI can be making in this area. I'm trying to make this quick. The second question, ethical challenges. Well, a big one I think is also around the interaction with industry. So in very different parts of the world, people have different opinion on working together with companies. Genomics England, from the very beginning, they did a lot of campaign and work sort of to express the value of working together with industry, together with pharma companies, to the UK population, because that was a fundamental part of what Genomics England was about, and explaining the benefits there, so that when people came on board and when they were giving consent, it was made clear that this data, yes, it's for health purposes and for improving the NHS and your diagnosis, but it would also be shared with companies. But there can be some tensions around that, right? But I think a lot of that is also to do with explaining the value of it in the beginning and educating the participants in the beginning. But obviously, data needs to be consented for use by companies. And if it's not, then it should not be accessed by companies. And having very strong governance and policy around access to the data, even when it has been consented for use by industry, to be very carefully managed on how industry is interacting with that data, who's using it, and all of that. And it's tracked very closely to make sure that you stay in line with the consent and the purpose which has initially been consented for in the beginning. So I'd say that's kind of an area where it can get challenging, but it's not insurmountable.
Okay. We actually have more questions, but unfortunately, due to limited time, we are running out of time, so we have to end this very interesting discussion. Thank you so much, Dr. Jacqueline, for accepting our invitation, and it's an honor to have you here. And we hope to see you in the future, to have many more.
I hope to come over again sometimes.
I hope
Hopefully
so.
see you in person.
Looking forward to seeing you in the future. I would also like to thank the organizers of this webinar. I would like to thank our college and UAE University, and special thanks to Dr. Ifat, Dr. Azhar, Mr. Ismail, Mr. Al-Sajir, Mr. Aminu, Ms. Bayan, Ms. Khadija Al-Balushi, and Mrs. Mariam Al-Nayadi for their contributions and continued support. I would also like to thank our wonderful audience for the active participation. Kindly remember to please fill out the webinar evaluation form in order to get your CME certificate. And if you have any more questions or you want to contact Dr. Jacqueline, this is her email and this is the logo of her company. So if you want to check it out online, don't forget to stay connected with us across the IPH social media platforms. These are our handles. And next webinar is actually on the 22nd October, and it is on behavior of motorcycle riders, presented to be presented by Dr. Kumar Sumit and moderated by Dr. Preeta. So thank you again, everyone, and see you next month. Thank you again, Dr. Jacqueline. Hope to see you soon. Thank you and take care.
Bye.
Bye.